Okay, good morning, afternoon, evening, everybody, wherever you are. Happy to see so many people online. Today, so my name is Jack Dekkers. I'm a professor at Iowa State University in Department of Animal Science, Animal Breeding and Genetics, and one of the co-PIs of the Agriculture Genome to Phenome Initiative, funded by USDA. Very appreciative of their support. All information about this project you can find at AG2PI.org. So today, the field day 17 already, we're going to talk about incorporating domain knowledge in genetic and genomic prediction and analysis models. So we've talked a lot in the past about the phenotyping we can do, high throughput phenotyping. We've talked about machine learning, AI, at presentations by the different AI institutes. So now we're going to talk about, is there domain knowledge that we can use to inform our prediction and analysis models? So, and it will be myself and Dr. Christine Diepenbrock. Christine is an assistant professor in the plant science department at UC Davis, University of California, Davis. She got her PhD from Cornell University and then spent several years at Corteva working on crop growth models and integrating them into genomic prediction. And before she moved to UC Davis in 2019. And so she's going to talk about the integration of crop growth models for whole genome prediction in plant breeding. So Christine, so as usual, put questions you have in the chat, and I'll scan those and I'll interrupt the speaker if it's immediately relevant. Otherwise, we'll just keep it to the end. So go ahead, Christine.
Okay, thanks very much, Dr. Dekkers. I will go ahead and share my screen. Does that look okay, Jack?
Yes, it does. Perfect.
Great. Okay, thanks very much. So as mentioned, I will be discussing our work to integrate crop models and whole genome prediction in plant breeding pipelines. We do have certain of the co-authors and collaborators also on this call, and we're hopeful to have a full Q&A with a larger group discussion. Great, looking forward, and what a fantastic initiative. Thank you for the invitation to speak within this venue. Okay, so our lab was established 3 years ago, and we do have this point of emphasis of developing integrated physiological and genomic approaches. We are specifically focused on doing so in food crops. We are specifically focused on doing so under conditions of abiotic stress, alongside more optimal or well-watered conditions in our case. And we are also interested in adding in some of our crop nutritional quality work to these frameworks so that we can be modeling whether it's yield and other agronomic traits and quality and compositional traits in tandem. As mentioned, we do have quite a heavy focus on crop nutritional quality. We conduct phenotyping of all throughputs of nutritional quality traits. We do dissect genes underlying these traits, or dissect the genetic basis thereof, and then we are implementing the results in genomics-assisted breeding strategies for the delivery of products at scale. We are looking as well, given that nutritional quality traits have not explicitly been valorized along the supply and demand streams for many of the crops that we consume. We are looking for kind of win-win scenarios between nutritional quality and other traits that might already be priorities, might already be selected upon in the breeding process. And finally, we are working to ensure that the nutritional quality that we are breeding into these crops does make it through to time of consumption, to ensure that we really are achieving delivered nutrition and those improvements are sustained through to reaching the consumer. Great, so this is an integrated set of approaches. I will focus today on the first, which is listed there in bold. And next, I'll lay out kind of the rationale for why that has been a point of focus for us. Great. So as we might observe, when traversing a given field experimental trial, we will see, different genotypes of a given crop progressing at a different rate through their growing season. One example of this could be, we see quite extensive differences in flowering time. For instance, if we are walking through a diversity panel or otherwise traversing a diversity panel representing global diversity of a given crop. So we might see a range of even a month or more sometimes in that flowering time. And that's just one example of a developmental time point, but we see quite a continuous range, including with regards to leaf appearance rate and other kind of more fine-tuned parameters. Indeed, breeding decisions often do operate at the level of individual genotypes. And I will use that term in this talk to refer to accessions or cultivars or individual representations of a given crop that would have distinct genetics. So really, if we can get this physiological trait information into the units of genotype-specific trait estimates or breeding values and or marker effects, this might really help us in integrating physiological traits into the breeding process in a routine and cost-effective manner. Of course, with the ultimate goal of accelerating our gains and improvement for yield, other agronomic traits and or other traits that are already priorities for breeders. I would say, it's not explicitly our goal. We don't just integrate physiological traits for the sake of it, but rather because they have really important relationships to some extent are underlying our final priority traits. So they are kind of means to an end, but they can really give us important information, as I hope to kind of convey today. Okay, so we have a poll question just to get a sense of who is present, and I'll go ahead and launch that, hopefully with success. Oh, that's the second question. There we are. Okay, so we will have two questions. One, the first is for those studying crop systems or plant systems, and the second is for those studying animal systems. You may or may not see both at once. Great, okay, this is very informative. Okay, thanks very much. Feel free to continue voting. It looks like we have quite good representation across the crop types. We did use the USDA Economic Research Service definitions of crop types just to try to ensure that we were spanning kind of the gamut there. But please let us know if we have not captured your system. I would love to know that for any future implementation of this type of question. Great, okay. And Jack, hopefully this is informative for your portion as well. I don't know that you can see these responses. Okay, great, thank you. We will record that and move on. Okay. I will end the poll now. Thanks very much. Oh, sure, I'll go ahead and share the results. How about it? Sound good? Okay. So here's the results there. So again, we have quite good representation. Let's see. Great. Okay. We'll continue here. So the first example that I will show is indeed in cereals, but we will discuss other crop types in future portions of this talk. So I thought to define a couple of terms just to ensure that we're kind of on the same page as I use them for the next 20 minutes or so. We do speak quite frequently within breeding efforts of the target population of environments. And this would be defined as the key environment types within the target geography for a given plant breeding program. So the geography for which a given program is breeding, we would think about the key environment types or production environments or settings in which these crops are being grown. We would also consider the frequencies of occurrence of these environment types. And Mark Cooper and team have a great set of literature on that topic. As I mentioned, I will use genotypes interchangeably with accessions or cultivars for the purposes of this talk. I will mention a half diallele at one point, and that would be when crosses are made between all parents of interest, but with the reciprocal crosses not made. And finally, when I mentioned managed stress environments, I realized that this might be more common in plant breeding, but effectively this would be an environment or a location year or site year in which we are applying a given stressor in a controlled manner for purposes of field experimentation. In our case, we have included a control treatment side by side with our stress treatment of interest. I will mention, this can be more, how do I say, more tractably conducted for, say, drought stress compared to high temperature stress. But thankfully, there are others doing fantastic work on kind of how do we incur a managed high temperature stress when we can have a control in a very similar environment. But that is what I mean by managed stress environments, just that we have these experimental stations or sites where we can impose a stress and keep that well-watered or otherwise optimal treatment side by side. I'm going to really look in a controlled way at the measure of that stress. This could be contrasted with what we would call a random stress that would incur or be incurred endemically in another environment. Okay, so this crop growth modeling whole genome prediction, I will spend several slides on this framework and one application thereof. And this framework has been described as a generalized linear model or GLM version of whole genome prediction in which the crop growth model acts as the link function. And this is from a paper by Charlie Messina and team. I would note that this team and colleagues have developed a series of papers that are highly informative, a mix of simulation and empirical studies that have really built up this framework. And the portion that I am describing herein is the application of this framework to a large breeding experiment in maize. It's a half diallele. And looking at kind of the opportunities that are afforded to us in that framework and kind of comparisons to classical WGP, as we would call it. So the way that this framework could operate would be that if we have, say that we have observed yield from a number of locations, and we could effectively fit this modeling framework backwards, where we run this observed yield values, we run this model backwards and estimate physiological traits. Now, Tom Tang, who is listed here, and team came up with a kind of a forward selection procedure, not too indifferent from kind of a stepwise selection model, where we can end up determining the final set of traits that will be in our model. In our case, we ended up with 4 traits that will be depicted in a bit here. But effectively, these would be genetic parameters or highly heritable, not largely susceptible to environment management or interactions thereof or with genotype. So largely genetic parameters. And we would, ideally, these would be factors that are underlying differential yield performance in the environments of interest. So those are 2 key criteria that we use, highly heritable and important for our priority traits. We can then get those regularized to marker effects, which is really helpful in the sense that, with genomic prediction, sometimes our goal is to predict untested individuals. So with everything at the level of marker effects, we can end up predicting for tested or untested genotypes, which is a strength of WGP. And then given that the crop growth model provides a means by which to integrate environment and management information, whether this be from a weather station or soil related parameters or irrigation or other management factors, say planting density, we can then run this model forward for tested and untested genotypes and tested and untested environments. Note that the assumptions of the crop growth model would still need to reasonably be met, and that there's various tests we would need to conduct to ensure that. We can run this model forward and end up obtaining predicted yield as a function of that crop growth model. So effectively, we have a couple of traits that we're pulling out and estimating at the level of individual genotypes, and then we're running back through this genomic prediction to obtain predicted yield. Now, in contrast, classical WGP would be going directly from yield to getting these marker effects and predicting breeding values, et cetera. So we end up with kind of an additional set of information or an additional set of layers that go in when it comes to incorporating the CGM. Okay, so in terms of this large maize breeding experiment that I will discuss, there were several environments throughout the target population of environments in this case. Many of these were well-watered environments. There were also managed stress environments, experimental stations in Woodland, California and Buluco, Chile. So the stress in this case was drought stress at flowering, which is a pretty critical period for maize in its development. And I will describe kind of the results across these sites hereafter effectively. So our predictive abilities was CGM-WGP versus WGP. These are cross-family predictive abilities. We'll start on the left side of the slide and we will spend some time on it and kind of walk through it systematically. So if we are looking only at well-watered data, we end up being able in all 4 quadrants of prediction where we have tested and untested genotypes and tested and untested environments. We observed with CGM-WGP predictive abilities of 0.79 versus WGP 0.74, and I'll go stepwise through the quadrants. So 0.61 versus 0.62, et cetera. So we saw pretty similar performance of these models performing roughly at parity with each other. Now, when we started to integrate data from those managed stress environments, or this abbreviation here would represent water deficit environments, We saw that the predictive abilities in CGM-WGP were maintained, so 0.79 versus 0.8, 0.61 versus 0.58, etc. Whereas those of WGP did end up suffering. So we did see 0.74 to 0.49, 0.62 to 0.42, etc. So CGM-WGP-A was performing at parity with WGP, even in well-watered environments, and B was able to maintain its accuracy, even if after that information from managed stress environments was incorporated. So these were pretty interesting findings for us going forward. So as I've just described this aspect here, we did also observe, and I'll note that we also checked within family predictive abilities, given that that could be of interest to breeders as well, and saw similar patterns as we did with the cross family, which is what was depicted on that previous slide. Additionally, we did note, as noted in the second point here, we really observed stable estimates for physiological traits in CGM-WGP when we were including multiple environment types in the training set. So when we had both well-watered and water deficit data present, our data from those environments present. We also observed that estimates were stable when estimating multiple traits simultaneously. And this included multiple traits for each of kind of yield potential and stress or drought tolerance. So this was an interesting finding that we were able to have multiple environment types present simultaneously, and we were able to carry out that stable estimation in multiple regards. Okay, so a schematic kind of what does this crop growth model look like, to look under the hood a little bit. We end up having a daily timescale crop growth model, and various sub-processes are represented therein, where we have the leaf number and canopy development taking place. We have transpiration, we have radiation use efficiency, we have certain environmental variables related to soil radiation, temperature, rainfall irrigation coming in, that interact with some of these genetic parameters and give rise to emergent properties. And by emergent properties, I would mean in this context and kind of a systems way of thinking, that they're a result of a more extensive set of the effects of genotype, environment, and management, their main interaction effects. So we do have this biomass accumulation and partitioning, where eventually we end up going into the reproductive phase and ending up with a yield and harvest index. So this is kind of a baseline level of crop growth model built upon by Mark Cooper and team, and by Muchao and Sinclair had done some foundational work related to this modeling framework. And here's an example from Sandra Huyn Trong, where effectively we ended up having, we can simulate this canopy development. And if we're interested in radiation use efficiency, or biomass accumulated per unit of intercepted radiation, we can kind of pull out any given submodule or subprocess within this crop growth model, of which we just viewed the schematic, and pull out a certain parameter therein. Again, we would be looking for what we would consider a genetic parameter, highly heritable, not susceptible to the effects of E and M, and or their interactions with G. So we then get that at the level of individual genotypes and get that to marker effects. So this is one example of a parameter that we could pull out to be estimated. One of the opportunities that was particularly of interest to me in this work, that ended up being, pretty interesting to consider. We can look, if this entire cloud depicted in gray in each of these biplots is the full half diallele, the set of the population of interest, we can look at the progeny of any given cross within that cloud, and take a look at kind of the physiological trait values as estimated in this modeling framework. So I would note that if we look at non-stiff stalk 8 by non-stiff stalk 5, this would be two parents being crossed to each other. And I would note that these are our elite parents, not particularly drought tolerant. We end up seeing that these progeny effectively have above average values for this senescence trait, as we call it, so leaf senescence response to water deficit. And it turns out that negative values of that trait indicate stay green, or lower values, in this case, are favorable for drought tolerance. So it turns out that this cross did not really end up producing individuals that have favorable values for that trait with regards to drought tolerance. So effectively not having the stay green trait. Whereas as we start to cross with more drought tolerant parents, again, we can kind of see how these progeny appear in germplasm space with regards to the estimated traits. So for example, some of these crosses, you end up seeing the progeny having those more favorable scores for senescence, or having that stay green trait. We can also consider other traits that may be of interest for drought tolerance, for instance, husk length. In this case, could be of particular interest to ensure that the silks are emerging from that husk and able to be successfully pollinated. So relates to the synchronization of that anthesis and silking. So kind of minimize that anthesis sibling interval, as we call it, or operate in a way that we can achieve successful pollination. So we can kind of look at which combinations of traits are really important for incurring improved yield performance under drought and stress conditions. Great, so we'll focus on that. We'll leave that there for now. I think that will be sufficient for purposes of today. But here's the list of the 4 traits that we ended up estimating in this process, and number of rings per year ends up relating to, as we think about the kernel rings on that year of corn. And of course, we may have multiple years in certain scenarios as well. But these were traits that ended up appearing to underlie the differential yield performance observed in this population. And of course, I will mention this is hypothesis generating as well. So it's really important, of course, to check these back against the observations of breeders, conduct further field studies to examine the biology underlying some of these findings as well. So this is a tool. It's not the be all end all, as we say. So we did note, and this is a finding that Charlie Messina and team had found in one of their previous papers as well. They had found that Cgm-WGP looked to have increased utility in situations of more extensive G by E interactions or genotype by environment interactions. We found specifically in the context of this paper that Cgm-WGP had a predictive advantage, particularly in instances of decreasing whole season evapotranspiration. So it did look like, under those stress conditions, we did see that predictive advantage of Cgm-WGP be more pronounced. However, I would note, as mentioned previously, we did not see a penalty incurred by having that crop growth model as the link function. So it turned out that Cgm-WGP was performing comparably with WGP, even in the absence of those stress conditions. So it may not hurt to have it be one of the standard methods or more mainstreamed going forward. Again, as mentioned, these physiological trait estimates were quite stable as we tested kind of different configurations of the model. And finally, this is in silico germplasm characterization to some extent. It is based on biological priors that were established for this population, or prior ranges for the mean for each of the parameters tested. But we did end up integrating Cgm-WGP as well as a gap analysis framework where we can look at attained or realized yield versus yield potential as simulated by the crop growth model. And then we can integrate that Cgm-WGP and gap analysis. They again have complementary strengths where the former can really look at early stages of the breeding process, potentially helping us screen large numbers of individuals. Gap analysis can help us integrate with some of the agronomic considerations and feed that information again into those early stages. So again, this is in the context of hypothesis generation. As shown in those biplots, I'm really excited about these opportunities to kind of look at the strengths and vulnerabilities of certain parental combinations. So whether we might be kind of exacerbating a vulnerability or complementing it, or maybe having redundant strains of the germplasm as evidenced by the parents and the crosses that we're making. So again, there can be a debate within genomic prediction, are we selecting parents or crosses? There's been great work by others on this topic, but I think there could be opportunities in either regard from this approach and set thereof. Okay, great. So we were curious as well among you all as attendees, the information types that you would most seek to integrate with genomic prediction. There are many types of domain knowledge. So let's go ahead and launch that poll to see what you all have in mind. And then we'll take a minute to read through this, so we'll give some time here. Okay, great. We'll give about 20 more seconds. Wow, okay. Let's see. Thanks very much, everyone. Hopefully you had an opportunity to vote if wishing to do so. I will go ahead and share these results. This is a very even spread, so that is excellent to know. Great, this is extremely fascinating. Let's see, okay, great. And I'll mention, our group has done some work, given our emphasis on nutritional quality and integrating information with regards to major effect genes into our genomic prediction models. I did opt to not focus on that work today, given that I think others have taken more sophisticated approaches since the ones that we did. We do have some active work in that area and look forward to sharing that at a later date. But yeah, these are very much in play and are really exciting areas that I know many of you who are here are also working on. So keen to interact with you all on that. Okay, great. Thanks very much. Close that out. Okay. Great. So I thought to describe a couple of other projects that we have that are earlier in their development. But we do have a couple of efforts. Jack mentioned the AI institutes that had come and presented earlier in this venue. And we do have a project through an institute that is based here on the UC Davis campus with collaborating institutions as well. So this is the USDA Nifa AI Institute for Next Generation Food Systems, led by Dr. Ilias Tagapoulos in computer science here at UC Davis. And we are specifically, our lab is involved in a project that is focused on sensing and modeling of leaf biochemical and physiological traits. So we have a couple of goals for doing this. Again, these traits are not, we don't just work on these traits for the sake of it, but rather because they are important to nutritional quality and to plant structure and function, as gives rise to our priority traits. So one of the reasons why we're interested in working on leafy greens in this context, so we've moved on to vegetables here, is, those leaves both are, of course, very important physiologically to the plant and are also being directly consumed by humans. So we have this kind of commonality of tissue in which we can examine questions of relevance to multiple priority traits in tandem. So we are looking to carry out and have been carrying out non-destructive and simultaneous sensing of biochemical and physiological traits. We seek to do this in entire breeding populations. We seek to do this for the entirety of our field trials across replicates and treatments. And we seek to enable an informed selection. These are decision support tools, and we are also kind of integrating this crop growth modeling, again, in a way that we don't seek to complexify how selection might take place, rather provide complementary information that is using some of the same data streams that breeders are already using and is using, again, the same functional units that are already in play in our breeding decisions. So we are looking in this case at both mechanistic models, which are similar to those that I depicted in the schematics shown previously, and AI-enabled models. And I'll show an example of that shortly for crop growth and development. In some cases, we're thinking more about broad-scale crop structural properties. Maybe we don't have to kind of build up the leaf appearance rate and the fine-tuned structure of the plant. Rather can look at, and there's been great work by other groups as well, to look at overall shape of the canopy or that type of thing and test how predictive those kind of more coarse models might be. Great, and we are aiming, to simulate both yield, however we define that in the crop of interest, whether it's marketable yield or another metric, and quality parameters that are, again, already valorized along the supply and demand streams. So we are dealing with multiple types of sensors and deployed via multiple form factors. We do have destructive ground truth measurements, which can tend to be a limiting factor, of course, of collecting this nutritional quality data via gold standard methods, as we call them. And finally, we're integrating high resolution genomic data into these frameworks as well. We would like to thank Dr. AG2PI and team, including Emmanuel Gonzalez, who's been doing a huge amount of work in the data examinations and analysis at University of Arizona, as well as Cybers, with whom they have been working. Great. So I thought to highlight as well one collaboration with Drs. Mason Earls and Brian Bailey here on the UC Davis campus. So Dr. Bailey has a really interesting framework for 3D biophysical modeling that they have deployed in various crops at this point. Depicted here is a figure from their paper, which is really an excellent read. And they have been really integrating their understanding of plant physiology into these models, and again, can look at crop structure and function in a really unique way from what I've been observing so far. They have depicted here the Stanford dragon modeled, and then a couple of different crops and physiological properties thereof. We are looking at integrating and deploying these frameworks. So AI-enabled sensing, 3D biophysical modeling, and pre-breeding and genomics. And we are specifically working on grain, legumes, and sorghum in that context now as well, grain sorghum specifically. And we have 3 members of our team who are working on this project. Dr. Sasum Lo is a cowpea breeding and genomics expert who is now expanding to other crops as well as Johnny Berlingeri and Earl Renario. So this really has been a collaboration across engineers, breeding and genomics experts, and experts in kind of use and analysis of sensing data. So one example that I thought to highlight, this is full credit to Drs. Bailey and Earls, and they have an interesting preprint. This one's in Grapevine, but depicts, a similar set of analyses in case you're interested, where they can, through use of multiple sensors, these are synthetic images created from Helios. So that 3D biophysical modeling framework, which is a software platform developed by Brian and team. Effectively, they can pull out different indices that might be of interest. They can look at different sensor information. And interestingly, as depicted in this last image, they can also create kind of photorealistic images from these synthetic images. And Brian and Mason have found a way to effectively, with learning, they can end up having labels already appended. So they can generate photorealistic images and have these labels. So this can help a lot in the annotation process. And this ends up involving a semantically constrained generative adversarial network in this case. So really excited to see what's coming next in these crops. Again, we're in the early stages of that collaboration, but we're really keen to be working with these experts in AI-enabled sensing and 3D biophysical modeling and integrating those with genomic prediction in particular. I would mention again, these are approaches, these are tools. Our end goals are still to inform the improvement of productivity and quality. Okay, and one reason why we are working on these different approaches, we have in mind to build up flexible prediction frameworks. Some of the challenges that we've faced in various contexts in our previous work has been, depending on distance away from the breeding station or the technology platforms that can be, or phenotyping platforms even that can be deployed in a given context, we may or may not have all of our data streams enabled in every field trial that we are conducting. So to kind of afford that additional flexibility, I just thought to place for your examination. So if say that we have 3 parallel work streams or data streams where we have sensing data coming in, we could predict biomass or other traits of interest directly from that sensing information. We've discussed genomic prediction selection, where, eventually with a well-trained model that we are also continuously updating, we could end up predicting biomass through use of genomic information as well, and the accompanying phenotypic. And then crop models, you can also simulate biomass in that framework. So, if all of these are enabled, we could consider ensembling across these at some level, or kind of letting the intermediates interface in some way, which I've kind of depicted here with the purple. So we could bring over information from sensing data, from extracted features therein, or we could bring in kind of simulated emergent properties, et cetera. So I've done this before where I show a very large number of arrows going between these, and suffice it to say, there are some really great opportunities for integration, but we also, the goal would be that any 1, 2, or 3 of these could run in tandem depending on the data that's available. And we can test, how many of them are needed to achieve a reasonable level of prediction. If some of these are not really giving us substantial added gains, then perhaps we can simplify as well. But that's the goal, is to really achieve maximum flexibility, but also make sense of the information that we do have to the maximum extent that we can. A couple of notes that I've listed here. This is kind of a thinking out loud, but has been a continuous set of considerations for us in this implementation. Would be that, again, I've mentioned these genetic parameters that would only, or at least largely, be a property of the genotype, whereas emergent properties would be emerging from a more complete set of these G, E, and M main interaction effects. Again, there's been great work by others, but we do have the, in the case of genomic-based relationship matrices, those give us different sets of information than hyperspectral relationship matrices do. Again, we could think about using both of those. In some contexts, we may only have one or the other available. So just thinking about what those matrices represent can be really important and affect how we use them, of course. And then I'll mention as well, so thus far, genomic prediction and selection have been based, mostly on genetics and genetic effects. Again, there are others on this call who've been doing fantastic work to try to expand that out to consider more of environment and management in their interactions. There's been work incorporating static environmental covariates. But I would mention, genomic prediction and selection have the advantage of being able to work with genetic effects. So that's very helpful. Whereas crop growth models and 3D biophysical models provide a means by which to integrate E and M effects. So these do, in that very high level nature, are complementary. And another nice thing about these crop models is they provide a dynamic means by which to integrate the environment and management effects, as they affect crop growth and development in real time per se. That aspect of the model may not be perfectly parameterized, and it takes a lot of careful experimentation to build up these models, thanks to physiologists from multiple decades now for getting these tools in place. But really, these frameworks can be complementary. They give us kind of a dynamic way to integrate the effects as they are incurred over the course of the growing season. And I finally would note, this talk is focused on integrating domain knowledge into our genetic genomic analyses. But I would note, the sensing data in the crop models, they could provide information that can be leveraged directly in prediction and breeding. So they don't necessarily need to go through genomics to be able to be used, but and indeed can be complementary and hopefully provide accelerated gains when we get all of these working in tandem. So I'll say we are, again, trying all of the kind of pairwise and three-way integration of these as well. Okay, so one example of kind of how this plays out in the course of a given project, Rebecca Dumonsky in our lab has been working on this effort. We have 2 field seasons of data now, which we've been very keen and are actively analyzing, where we, in this case, are looking at well-watered and managed pre-flowering drought conditions. Part of why we're interested in pre-flowering drought would be that grain number in sorghum is determined in that window, in that developmental stage. So, and again, excellent work by others has found kind of the importance of grain number as a yield determinant in sorghum. So we're taking a look and trying to model, again, this critical yield component, its effect on final yield in diverse material. We are carrying out nutritional phenotyping, which takes quite a lot of time, even on high throughput platforms. And then, carrying out our genetic genomic analyses, we are also seeking to pair those with the crop growth modeling. And again, this is kind of one of our first large scale attempts to look at both productivity and quality in tandem within the context of crop growth models. Now, I would note, sometimes we want to think about when does it make sense to have a standalone model that does not run in tandem with the crop growth model, and or when might we want a more detailed time course through grain fill. So we're just really doing a more mathematical model of how these compounds are accumulating and being retained versus, again, pairing this with a crop growth model. One point that I would just note is that some of these crop growth models do consider or do have baseline considerations of grain nitrogen, for example, already embedded for various reasons that I won't go into. But there are kind of nuggets there present in some of these models off of which we could build. But that is an active consideration where to what extent do we want this paired? Another reason to pair them could be, if you're interested in kind of how senescence and grain protein end up relating to each other as one example, or again, leaf carotenoids versus the carotenoids that are available for human consumption and for absorption and used by the human body in animal systems as well, I would say. Yeah, there are reasons, of course, for different types of models, and it depends on the question that we're asking. But this is one framework in which we are deploying these types of methods and getting them integrated into the quantitative genetics that we've been doing all along as well. Okay, so here's a summary for my portion. I'm very keen to hear Jack's thoughts. But effectively, crop modeling and whole genome prediction, we have found them to be highly complementary. They do inform multiple stages of the breeding process, but namely they can help us out early in that process with scoring a large number of individuals, which can be a limiting factor in certain programs and contexts. We are looking to work on both productivity or other agronomic traits that are already prioritized and nutritional quality. We are looking to breed for the target population of environments and really be considering actively those multiple environment types in active conversation and collaboration with growers as well. Of course, these models do not replace phenotyping. The experimentation and phenotyping thereof will continue to be critical. Again, genomic prediction and crop models end up being complementary for reasons mentioned. Again, we're seeking to use the same data streams already in use by breeders. Crop models, we're looking to examine both structural and functional traits. We're looking to integrate those environment and management effects and get the main interaction effects of G, E, and M as well dissected as we can, while also not over parameterizing. There are limits to our domain knowledge, so we want to make sure that we're building up these models progressively and not over relying on them. And finally, there's the final portion there. So we can really start to look at these physiological traits, and I would add biochemical traits as well, as we're now integrating. And we can consider some of these main interaction effects both in the estimation steps and in the prediction steps. We see utility in both of those steps. So I will stop there. And thanks very much. I will look forward to discussing. Okay,
thank you very much, Christine. Very interesting presentation. So we have quite a few people online, so please put your questions in the chat. And we have one question already from Katie Navabi. With the vision of shortening breeding cycle using genomic prediction strengthened by sensing data, how do you suggest it be applied in the breeding pipeline when the test population needs to be planted and sensed or phenotyped?
Yes, that's a great point. So yeah, if we're, genomic prediction, and selection, one of the benefits could be that we could speed up that cycle and genotype very early. Yeah, I suppose, in some sense we could reduce the number that need to be sensed for the entirety of the growing season. There's been work to see how well can we predict later stage phenotypes from early stage phenotypes. I think that is a really important consideration. So yeah, it may be that, our accuracy is improved or our correlation with the traits of interest is improved. There might be other ways within that response to selection and breeder's equation that end up being able to be improved. It may not be the cycle time to as much in this situation. Hopefully we can improve all of the above, but it's a great point. We do need whole season phenotyping for some of these traits.
So Christine, I have a question. So you talked a little bit about these crop growth models and developed by physiologists, but could you say a little bit more about the origin of those models and their original purpose?
Sure. Yes, and I'm sure there are others on this call more qualified than I am to answer that. But I would say, there's been broad development and deployment of crop growth models by many experts in the field who, they've been deployed quite a lot in agronomic context as well, and considering effective management on different genotypes, really detailed considerations of physiology of these crops. However, there have been, to my understanding, and from what I've been observing and seeing in the literature, less examinations of large breeding populations, including for the reason that it can be really tough to get these models parameterized for large populations. So this model that Charlie Messina and Frank Techno and team have been developing over time gives us a way to scale up their breeding populations through kind of the sampling algorithms that they've enabled and the careful experimentation through which they defined relevant priors.
So there's quite a bit of work that's needed to validate these models before they're being used.
Yes. Yeah, and we were very careful to try to show that in the paper from which I'll show this again briefly. The plant physiology paper, part of the goal of that paper as well was to be sure that we're laying out kind of what are the steps involved in training parameterizing this model. So, not on that one, but this paper here, we really tried to lay out kind of what would be the steps in building up that model.
Yeah. Another question from Nimai Senapati. How to establish the marker effect within the WGP CGM?
How to establish the marker effects? Sure. So yeah, I suppose this would be back to linear algebra. Once you have the physiological trait estimates and once you have the DNA sequence profiles of the genotypes in your training set, you would end up then getting those back to marker effects in that manner. So it is kind of, one of the sets of algorithms that we're more familiar with regards to genomic prediction at that point. Effectively, we're fitting marker effects for those intermediate traits rather than for yield itself.
Yeah, and I'll show that in my presentation.
Perfect. Wondered if you might.
Yeah.
Great.
So.
And Jack, I would like to, hopefully there's enough time, I would like to just acknowledge this co-author team from the CGM-WGP, as well as, Corteva AgriScience and the management teams thereof who did support this, the maize large breeding experiment work that I mentioned, a large number of the AI Institute team collaborators, the Nifa sorghum project, and then as well as Summit Zimbabwe project team with which we've been thinking about some of these questions of abiotic stress tolerance. And finally, the UC Davis Department of Plant Sciences.
Okay, so one more question online, actually two more questions online. How to integrate crop model parameters and SNPs? Is there any requirements for the crop model parameters?
Any requirements? Let's see. Yeah, I'm not entirely sure that I have grasped. So I suppose I would say, we do want to ensure that these are parameters of the genotype, because if there's environmental information kind of confounded in there, we would not be able to have accurate effect information for the SNPs. So I'm wondering if that question might be somewhat similar to the previous in the sense that we are trying to get back to, individual markers and effects thereof.
Yeah, that's right. Final question, and then we'll have to close it off. Why use existing labor-intensive physiological traits as opposed to the new AG2PI traits to parameterize the crop growth models? I suspect until that is done, it is tough to extend to whole breeding programs.
Sure, yes, and thankfully there has been work by others, including in sorghum, on this topic. So again, this is a community effort, and we're looking forward to discussing as a continued discussion. I would say all of the above have their place. That's part of why we've pivoted to some extent in these latest projects towards some of the 3D biophysical models. We're still using mechanistic models and can even compare the two types. But to be able to directly, as kind of came up in the poll question, to be able to directly input sensing data in a meaningful way that helps us in training our models for diverse genotypes, that is very much of interest. And I would say as well, that relies less on in silico, how do I say? Effectively, we can have more of the emergent properties captured if we can integrate that direct sensing information, and perhaps even can pull out some of the genetic parameters as directly observed, which kind of changes how we do that modeling framework that I showed. So yeah, effectively, the more we can observe, the better. And it's really nice to have that information and be able to conduct feature extraction and or use some of the sensor streams directly. So I am all for it. We're working on the implementation, I would say.
Okay, there's another question, but we'll leave that to the end.
Okay, sounds great.
And so thanks.
Cool, thank you.
Christine, and Christine will stay on, so we'll come back with questions which can be for both myself and for Christine. So I'm going to share my screen. To talk about how some of this is developing in livestock, and I'm not the speaker here because I think I'm the best person to talk about this. There's a lot of experts in the area, but I was just a person who was easy to twist my own arm. And hopefully I know enough about it that I can stimulate some discussion and relate it to what's happening in the plant side. Okay, everybody see my screen?
Yes, looks great.
Okay, let's see, I'm gonna, 1 second. Okay, so integrating biological models into genomic evaluation of livestock. And mostly I'll talk about what we're doing in pigs. We have a grant that I'll talk about from Nifa to develop some of these models to integrate a pig growth model into genomic evaluation for pigs. So I'll give some background and then I'll go into that project and some additional challenges that we have. And I have to say that we are not nearly as far along as what Christine described for several reasons. So a typical growth curve. So in pigs, there's a grow finish side, the grower side, and that's what I'll talk about. Typical growth curve of a pig or of any animal is this sigmoid curve, and often we fit what's called the Gompertz curve to that. The Gompertz curve was initially proposed by Benjamin Gompertz, not for modeling growth, but actually for modeling risk of mortality in humans. But it also works very well for height, growth, and so on. And it has 3 parameters. A is the asymptotic body weight in this case. B would be related to B and C both relate to the inflection point, where that occurs and what the rate of growth is at the inflection point. Now, unfortunately, in growing pigs, we send them to slaughter before they, shortly after their inflection point. So we don't really see the remainder of the curve. Although there is an interest to estimate mature weight, especially on the female side, where females are staying in the herd for multiple farrowings, multiple birth periods. Maintenance requirements of heavy sows are a lot larger than of lighter sows. So it is very much of interest also what the mature weight is. But based on the data we're getting, we are not able to directly estimate that. This is what it looks like for a group of pigs from one of our industry collaborators, Topix Northfin. So fairly linear. I mean, you can see some curvature. And so this already points out a challenge that, well, how are you going to estimate this nonlinear curve when your data almost looks to be linear? Because it's linear, what has been done in practice is just, take a weight at 75 days, take a weight at 180 days, just before slaughter, and then just calculate average daily gain. And that's an important trait that selection is based on. Then we're also interested in feed intake. It's one of the largest cost component of livestock production is feed. This shows the green line here shows a typical feed intake curve. Now, feed intake is hard to measure on an individual basis. In practice, animals are housed in pens. You could measure pen feed intake, but for genetics, we need individual feed intakes. And so breeding companies, for many years already, have used these electronic feeders where the pig has a transponder in its ear, goes into this race, and then it's recognized, and then the amount of feed eaten is measured. Very messy data, because if you put a sophisticated piece of equipment, electronics in a pig barn, you know what can happen. Lots of things can go wrong, and so lots of editing needed. So also that has more or less this Gompertz kind of curve, and this is from that same population, the individual feed intake data, very messy data, very noisy data. So what people typically do is then just calculate the average daily feed intake during this test period, and that's a phenotype that is measured. Another trait that breeders are interested in and the industry is interested in is meat quality, particular back fat thickness. We've been aiming to breed pigs that are leaner. To measure back fat, you have to slaughter the animal unless you use ultrasound. And ultrasound technology coming from the human side was employed several decades ago in the swine industry and also in the beef cattle industry to measure back fat thickness on the live animal. The company here in Ames that markets some of the software to read the images, so you can measure back fat, so the thickness of the fat. You can measure loin eye area, so the eye of the loin. And you can also, if you take a different orientation, you can measure intramuscular fat by analyzing the pixels in a square. And so that's being employed in the industry on a regular basis. And of course, that's needed in a breeding program because you don't want to slaughter your breeding animals. So a typical back fat curve would look like this, but because it's quite time consuming, it's only really measured at the end or just before slaughter. So that's where we get back fat. So then the standard traits are average daily gain, average daily feed intake during that test period, back fat, loin eye area, and intermuscular fat. Now to talk a little bit more how this fits into the overall strategy, it's important, especially for those on the crop side, to realize that a breeding program in livestock typically looks quite different than in plants, and I'll make a comparison more directly in a little bit. But the selection takes place in these purebred nucleus programs, segregating populations. So we typically, in pigs, we have a sire population, a sire line, and two dam lines that are crossed to produce crossbred females that are used for reproduction. They're crossbred to benefit from heterosis on litter size and other reproductive traits. And then the final market pigs are produced by crossing these crossbred females by what's called a terminal sire line. And that also drives selection in these different lines. So on the sire line, you're not interested in litter size because the litter size is determined by the female. So you're interested in growth rate. Feed conversion is very important. Again, going back to the cost of feed, and then different aspects related to carcass quality, meat quality, and to some degree also male fertility. On the female side, we have the same traits, but then litter size and longevity of the female, of the sow, becomes important also. So one way that sort of related to the heterotic groups in corn, although there's more difference in what the two sides of the pyramid of the structure are selected for. Also, when I talk to plant breeders and I ask them what's your breeding goal, they say, well, the three traits: yield, yield, and yield. In animal breeding, generally, it's much more diverse in terms of the number of traits that we need to improve, and that becomes important also. Just to contrast that with a plant breeding program, and this is a graph from John Hickey. So plant breeding program is almost an inverted pyramid, right, where you narrow down the genetic base as you develop your varieties that are marketed. So here, in animal breeding, genetic improvement really takes place at the top of the pyramid, and then we have the product development phase in terms of the crossing. In plant breeding, the genetic improvement phase and the product development phase, they're sort of intertwined. At least that's the way I would look at it. That also brings up some challenges, because most of the phenotype collection is up here in animal breeding, where in the purebred animals, and typically under very high biosecurity, because you don't want a disease outbreak in your germplasm herds, because you wouldn't be able to sell the germplasm anymore. So it's a very unique environment. On the plant side, these multiple environments and multiple crosses are made, and so the data that's collected is typically across many different environments, which of course helps with dealing with G by E. So on the animal side, and this is very much for swine breeding and poultry breeding, to a lesser degree for cattle breeding. When we focus all the selection based on purebred data in this high health environment, we're not necessarily improving commercial performance as well as we can. And also, there's limited disease here, so it's very difficult to select for disease resistance. And also, this is selection on purebred performance, not selection on crossbred performance. So there's lots of challenges with that. The importance of that can be measured by, if you look at the genetic correlation between the same trait measured in the nucleus on a purebred animal and at the commercial level on the crossbred animal. So we call that the RPC, the correlation between purebred and crossbred performance. This is a review from Yvonne Wintjes and Mario Calles from Wageningen. And for different traits, quite a range of estimates. Many of them being substantially less than 1. And especially fertility here, so that's on the maternal side, the average correlation being 0.6, which means that if you select on performance in the nucleus, only 60% of that is transferred to the commercial level. And it's been shown that these genetic correlations being less than 1 is a combination of G by E, so different environment, but also G by G, so purebred versus crossbred. Now, there's ways you can get around that. The traditional breeding solution has been to, go ahead and collect data at the commercial level. But then, of course, you have to feed that back into the nucleus using pedigree, which is typically very difficult to do in a commercial farm to track pedigree. But it can be done. It involves lots of logistics, and that has resulted in what's called recurrent reciprocal selection, or combined crossbred and purebred selection, where you use data at the commercial level on crossbreds, but still also use the purebred data in the nucleus, because that's still also informative. But very costly in terms of logistics, requires pedigree, and it also ends up leading to higher rates of inbreeding because you use a lot of family data in your selection program. With genomics, we have a way around that. We still need to collect phenotypes here, but we can also collect DNA and get the crossbred genotypes on the crossbreds, and then put that, use that as training data, and then the marker effects can be fed into the nucleus breeding program, and so select animals at the nucleus based on their genomic breeding value for crossbred performance in a commercial environment. You can do that both on the male side and the female side, and that's being used. Still lots of limitations because there's not one commercial environment. There are, and this is a paper looking at heat tolerance. So looking at a range of, I think this is temperature, range of temperatures, and the genetic correlations across this two-dimensional surface. You can see that some of these correlations, they get pretty low. So even in a commercial environment, you know, temperature ranges are quite different, even though there's environmental control to some degree. Temperature difference and humidity are quite different across the country and in different countries. And that leads to G by E, and you can use reaction norms to fit that and categorize that and identify animals that are more or less heat tolerant. So what can be done using mechanistic models? And there's been quite a history of mechanistic modeling in animal production also. A lot of that is driven by nutrition. Nutritionists trying to determine how to best feed an animal. That's the basis of the models that we're using. But again, similar to what Christine talked about, we're aiming to identify, based on models, underlying latent variables or physiological parameters that are less affected by G by E and less affected by non-additive genetic effects. And there's a book that came out in 2006 that documented a lot of these models, and a paper by Andrea Dushel-Wilson and Peter Knapp and Brian Kinghorn talked about sort of the difference of what can be done with these mechanistic models compared to what we do typically. Typically, what we do is we record phenotypes in different environments, in the nucleus environment, in the crossbred, the commercial environment. We do multivariate BLUP, and then estimate breeding values, not only for purebred performance, but also for commercial crossbred performance, and use that for selection. And they described that using these models, you can use them as for simulation, and then basically, given the genetic potential of underlying variables for growth, efficiency, and robustness, using that mechanistic model, you can predict or simulate phenotype, and their idea was then to invert that model to, given the observed phenotypes, aim to get the estimated breeding values for growth, energy, efficiency, and robustness. At that time, the success of that was fairly limited, in part because of the statistical methodology like Bayesian methods and MCMC weren't available, and also in part because genomics wasn't available. So, but they did realize that there could be very many potential benefits. So, because if you are able to find these underlying traits that are less affected by G by E and G by G, you can predict across environments, across breeds, from purebreds to crossbreds. You can use that to optimize phenotyping strategies. So find where should phenotypes be collected, on what animals, address what if questions, what happens if I put this animal or its progeny in another environment, and also predict potential limits to selection. And I say that there's no evidence that we are reaching limits. This is an example of in broilers, in chickens, 1957 genetics and 2005 genetics. And these birds, they're all 7 weeks old. So huge, and a lot of this is due to genetics, genetic improvement. So huge improvement has been made over the past decades, but there's concerns, well, are we reaching limits? And there's a paper here that suggested, well, maybe we are reaching some biological limits, physiological limits. And we really, to recognize, with our quantitative genetics machinery, linear models, we're not able to identify those limits until we have hit them. And so biological models may also help us inform what are the bottlenecks in terms of further improving traits. So I mentioned that in animal production, there's been a lot of work by nutritionists in particular to develop these nutritional mechanistic models of growth to develop nutritional strategies, most cost-effective, least cost diets and so on, while still getting maximum growth and leanness. And this is an overview of one of those models. The model that we actually used was developed at INRA in France by Jaap van Milge and others. And it has feed intake as an important trait that can be modeled, that is then digested. So you go from gross energy to digestible energy to metabolizable energy to net energy that then can be used for the different processes of protein deposition, lipid deposition, but also maintenance and activity and the cost of protein deposition. So there's quite a number of energy sinks that need to be considered in these models. There's the energy side, but then there's also the amino acid side. There are essential amino acids that mammals need that are not produced by the animal itself, like lysine, and that's another important aspect of these nutritional models. Now, question, I'll launch a poll just to get everybody thinking. Let's see. Oops, I didn't want to do that. Okay, trying to launch the poll. There we go. Okay. Hang on 1 second. Okay, so this is really the question about why does an animal grow? Or why does an animal eat? Does an animal grow because does an animal grow fast because it eats a lot? Or does an animal eat a lot because it grows fast? So what's the chicken? Is the chicken or the egg? What came first? So questions are, how fast does the pig grow? How fast the pig grows is driven by how much it eats. So feed intake potential drives growth rate, or growth potential drives feed intake. So how much an animal eats is driven by its potential for growth, or a combination of the two. So I'll wait a few minutes and okay. Got about half of the votes in. And that's actually a question that relates to humans also, right? And some of you may be experiencing. Okay, I'll close it here and let's see. Share the poll. Nicole, can you share the poll? Okay, it's shared now. So yeah, most of you think it's a combination of the two. And it's actually interesting because nutritionists don't agree on this. And physiologists don't agree on this. And it's probably a combination of the both. But it sort of also illustrates where we are in terms of knowledge of biology. And of course, it depends on the nutritional strategy. I was assuming here that it's ad libitum feeding, so it's interesting. So I think, yeah, I'm on the side of most of you that it's probably a combination of both. Okay, I'll close this. Okay, so so with that, let's see. Okay, with that in mind, we launched a project that was funded by, actually, let me go back one. It's funded by USDA Nifa to develop a Bayesian hierarchical model to integrate a pig growth model into genomic evaluation of pigs. Postdoc we had was Haipeng Yu, worked a lot on that. Rowan Fernando, who many of you will know. From the nutritional side, we had Jaap Vermillige. Andrea Wilson from the University of Edinburgh has done a lot of modeling, also on the epidemiological side, and these types of Bayesian hierarchical models. And then Egbert Knoll from Topix Norsvin. So they're one of the breeding companies, and we were able to utilize their data for some of the work that we're doing. Still a work in process, and we basically went through steps, and I'll lead you to the steps that we went through. The data we had was from almost 4,000 purebred boars, so we are still looking at one environment, a high health environment, purebred animals. The next step would be to expand that to other environments, but we have body weight and daily feed intake data during that test period, and they were also genotyped for 24,000 markers across the genome. From that, then we have daily body weight and daily feed intake over that growth period. We have the SNP genotypes, and from that we then we predict latent variables, which I'll explain in a little bit what we chose as the latent variables, and then have a link function, and the link function is an aspects of the in-rapport model, this mechanistic growth model. In this case, we only considered body weight and feed intake, so we didn't consider fat deposition yet at this point in this first step. The model looks like this, where we have basically a Gompertz model for body weight, to model body weight with 3 parameters, which become the latent variables. And then feed intake is modeled as a function of body weight. Okay, so the body weight, expected body weight goes into here for feed intake, along with some other parameters, latent variables. And so here you can see that we use this two-way relationship, that feed intake is determined by both body weight or growth, but also by some additional parameters of the animal. So the latent variables we're using are age at 115 days, a shape parameter, which relates to the inflection point, and also body weight at 65 days of age. And we still have to do a lot of work to determine that these are the right latent variables, that they're not affected by G by E and by G by G. We're not at that stage yet. And then for feed intake, we have 2 levels of feed intake at 50 and at 100 days as our latent variables. And again, we're not arguing that these are the optimal ones, but those shape parameters then drive the mechanistic model to predict feed intake and growth rate and related to the observations. On the genomic prediction side, so then we have a whole genome prediction model, the standard one, by using marker effects and also contemporary groups to predict the latent variables. And these are fitted as covariates because we want to get at the additive effects of these latent variables. So this goes back to the question that Christine got, well, how do you get the marker effects? Well, you get them by estimating or analyzing these latent variables in terms of marker covariates. And let's see, yeah, and then it gets put through the mechanistic model so this becomes the link function to link the SNP effects to the latent variables, and then to the observed phenotypes, which in this case are body weight during this period and feed intake during this period. So the body weight and feed intake you've seen already. So what we did for training, we took the older animals, and validation, we took the younger animals. And there are some of the prediction results. So bias, one is the estimate is unbiased, and accuracy. And of the latent variables. Now, remember these latent variables, at least some of them, we don't directly observe. And so in a normal model, in linear model, they would not be able to be estimated. But in this, by using them as embedded variables, we can estimate them, and then also using the Lagara and reverter method of part-whole regression, we are able to estimate their accuracy. And also we compared it, or Haipeng compared it to how good are the estimates of the standard traits, which in this case, we're looking at average daily gain from the growth model compared to the multi-trait or the bivariate linear model, where we analyze average daily gain and we analyze average daily feed intake. And you can see that it doesn't do any better. It doesn't do worse either. But that's not too surprising that it doesn't do much better, because we are looking at one environment and they're purebreds. So yeah, we don't expect to get a lot of benefit from the growth model under those circumstances. Now, one thing we can get from the growth model that we cannot get from the standard multi-trait model is our predictions of mature weight. Okay, the data are here, and average daily gain is over that period, but we are actually predicting out to mature body weight. Question is how accurate they are. This extrapolation, and how accurate they are will obviously depend on how good the model is. And some of these accuracies are given here. I'll skip through that. And also for feed intake, we're able to predict mature feed intake, okay, and accuracies. So our next step was to incorporate fatness or lipid deposition. And to do that, we were actually able to utilize some very unique data that Topix Northfin has. Now, we could use the ultrasound back fat and loin-eye area and so on, but they're using on their nucleus animals, on their boars, they use a CT scan to be able to measure detailed composition on the live animal. And they can actually look at different organs and different parts of different cuts of meat, basically. That we didn't look at that. We just are using lipid mass, total lipid mass, and total protein mass and back fat, because those are what are the key sinks, energy sinks, and amino acid sinks in an animal. So we have longitudinal body weight, longitudinal feed intake, and then we have the CAT scan results just at just before slaughter, at only one time point. So this is what that then looks like. Now we have a number of additional, now we are formulating it in terms of net energy intake rather than feed intake. But we have body weight different times, and latent variables relate to the body weight curve and the feed intake, and also protein deposition as an underlying variable. And again, the data we have is body weight over time, net energy intake, and then carcass composition at just at one time point. And then we again have the SNP effects as linear covariates, as well as contemporary group to as far as our ultimate outcome that we can use for genomic prediction. I should say that also the whole genome prediction model, so here we have 6 latent variables, is implemented as a multi-trait genomic prediction model. So considering the correlations among these latent variables, which are also estimated. So the mechanistic model is now a little bit more complex. We have body weight that derives net energy intake, although there's some other parameters there also, but primarily net energy intake is driven by body weight. And then we have the energy sinks, which is protein deposition, maintenance, and lipid deposition. And lipid deposition can then be translated to back fats. And so this is our mechanistic model and nonlinear equations that are behind that. We're still early on with debugging this part of the program, so I don't have any results yet, but that's where we are at right now, to consider both feed intake, body weight, and lipid. No, we can go a lot further and we need to go a lot further with that. We only, at the moment, we're only looking at the energy side. There's the essential amino acid side that needs to be incorporated. Looking at the growth of the different parts of the body, the different cuts of meat, if you will, because they differ in value. So ultimately, we hope to be able to get to a stage where we can focus on individual cuts of meat and how they are grown over time, or what the underlying parameters are, but we're not even close to being there at the moment. Then there are models related to immune and stress response. There's lots of factors that impact the animal, cause stress, and result in the need for immune response, which is costly. That side needs to be incorporated also. And there are models that are available there that would have to be integrated to accomplish that, and some of, as well as epidemiological models. So if you have a contagious disease, and if you want to model, you can't just limit yourself to modeling an individual animal. You have to model the herd and the environment, and how the pathogen is multiplying and spreading in that herd. So epidemiological models have to be integrated. So yeah, lots of opportunities, and the choice of latent variables that are not affected by G by E or G by G is still a very much an unanswered question for our models. These mechanistic models, they can get very detailed. You can look at individual pathways. You know, the Krebs cycle. You know, in principle, everything can be modeled. Do we need to go that deep? Probably not. But models are available. And if you look at humans, there's a lot of interest in develop from a medical side to develop mechanistic models of human development, and you know, from the cellular level or the gene level to the cellular level to tissues to organs to the individual as a whole, to try to model all of that. Again, I don't think we have to go that far, but there's a lot of models that are being developed in other fields, and especially, of course, with human medicine, a lot of funding that is available to develop those models. Now, this is my last slide. Hope to have given you some insight in the trajectory we're on, and lots of work needs to be done yet. Both on developing the models and integrating them and applying them to different environments and different populations. I want to contrast it a little bit by machine learning, and Christine talked a little bit about that. We've had all the field days about machine learning and AI, you know, it's a cartoon, and you know, there's a lot of interest in using machine learning, deep learning for complex trait genomic prediction, and you know, it definitely has its place. Let me launch another poll and ask you a question about this. Let's see, not this one. Okay, so you should see the poll. So what's your feeling about machine learning and AI, perhaps compared to other domain knowledge from other domains? Is machine learning and artificial intelligence, is that going to be the future of genomic prediction in plant and animal breeding, or not? Is it some combination of the two? Give you some time to answer that question. Okay, we got most. I'll end it here because it's already showing that most of you think it's a combination, that it's not, whoops, I didn't want to do that. Okay, so most of you thought that it's a combination, that it's not all, well, some feel it is definitely going to be machine learning. Others, but most of you feel that it's machine learning has a part, but that's sort of the way I feel about it also. Just another, just to get things going. So in was able to find a quote from, it goes back to 1865 in an editorial in an electrical trade journal. There was the time when, let's see, when electrical engineering and the whole field of electricity and electronics was developing, and there was an editorial. Somebody remarked that said that we may remark that of late years, the experimental facilities for obtaining definite ideas on the subject of electrical phenomena have greatly increased, and then continuing in electricity, there's seldom a need of any of mathematical or other abstraction. So this person was really arguing against using math to study or to develop electric equipment and electronics. And he or she, I'm not sure whether it's a he or she, although the use of formula may in some instances be a convenience, they may for all practical purposes be dispensed with. Now, I think if you talk to an electrical engineer nowadays, they would strongly disagree with that. There's lots of information on the basics of mathematics for electricity and electronics. And if you don't use the right formula, things can go very bad. So just putting that in context to what we're talking about here, sort of using machine learning and deep learning, mining the data for genomic prediction versus using quantitative, let alone biological models. I'm hoping we're not going to get into this situation that somebody's going to say that in a genetics journal that we have lots of tools that are being developed for machine learning data and statistical predictions for breeding, and that we really don't need mathematical models of biology. I think personally, I think it has to be a combination of the two, and the two have to go hand in hand to really make progress and get to G2P. So with that, I'm going to stop sharing and ask for questions. I haven't been able to follow the chat, but let me let's see. Okay, question from Guillermo. Why should we expect underlying physiological traits to be less affected by G by E or G by G? That's a good question, and maybe Christine can chime in on that also. I think we do see when we look at even gene expression traits, we typically find higher heritability, or there are the expression of some genes has very high heritability and limited effect of G by G. Not sure about G by E, but the idea is that the closer you get to the underlying physiology of the animal, the inherent physiology of the animal, the less it will be affected by G by E. Christine, do you have thoughts on that?
Sure. I suppose I'll say I think my answer would be somewhat similar. But when we get down to the level of certain sub-processes, that some parameters remain more constant than others. So yeah, these intensive experiments can help us to underlie those or identify those parameters that really are more truly genetic. It may not be perfectly genetic, but at least substantially so.
Yeah, but finding those, I think, is the key, right? And certainly the ones that we're using, they are not the ones that we should end up with, because they are very much affected by G by E. But we use them just to get the model developed, and then move into identifying which one. Are there others that are more useful in terms of their underlying genetic effects? There's a question: if the mechanistic modeling can be used to identify or determine the growth limit, then should selection breeding not be done? Then should selection breeding not be done anymore, since we've reached the maximum for some traits? And well, if we have reached maximum growth, there's still, it doesn't mean that we cannot improve the animal anymore, because we can still improve efficiency, right? So there's still other aspects of the production system that are important that can be improved. But I don't believe we'll reach that limit, I think it may lead to some. I think if we can anticipate our bottlenecks in terms of improving productivity through these models, then hopefully we can steer the ship better in the right direction to avoid these bottlenecks. And then also these traits are not independent, and no, they're not independent. And in our genomic prediction model, as I mentioned, we are modeling them as multiple trait with covariances. And I think, Christine, the same is done in the crop growth model.
Well, I wouldn't. So the traits are estimated simultaneously.
Yeah.
That would be the extent of it.
Yeah. We actually explicitly estimate the variance and covariance structure at the genetic level and the environmental level. Related to my first poll, plant scientists do not fully agree that photosynthesis is source limited or sink limited, which parallels the animal question. Seth's thought is that all plant evidence is towards sink limited. So in other words, that I think, Seth, you're saying that yield is the ability to grow is the limiting factor rather than the ability to absorb nutrients or sunlight. Do I interpret that correctly?
Seth left the call. He had to leave.
Okay. Well, all the plant breeders, do you agree with that?
I think we'd have to think through that differently depending on the crop, potentially.
Okay. Let's see. Okay, so then a question about the accuracy of prediction. Although the accuracy of prediction was the same for the growth model and the multi-trait model, but the ranking is the same. That's a good question. I think Haipeng has looked at that. Haipeng, are you still on?
Yes, I'm on, Jack, but I don't think I look at that, but I can check this, yes.
Yeah, okay. So that may very well be that the accuracy is the same, but the correlation between the genomic predictions of the two models may not be one. That's a good point. Okay, so let's go back to some of the questions from that we left at the end of the first talk. What other traits and yield can we predict with the crop growth model?
Sure. So yeah, that seemed to be related to the Cgm-WGP. There are other traits that are modeled within that framework. Whether they are the output trait of interest versus being an intermediate is another matter. So in terms of, let's see, other examples of successful predictions using Cgm-WGP. I would say the Cgm-WGP as a framework is the terminology that the team at Corteva AgriScience and their colleagues have used for that method. So I would say, there is other work to try to start to incorporate some of this mechanistic or other domain knowledge into WGP. So I would say, including among people on this call, there have been some successful examples of starting to test, can we outperform GBLUP? Sometimes we can't, right? That's been a comparison of models has been a kind of a long-term topic in this domain. So yeah, unfortunately, I think, expanding that framework beyond yield is something that has been of active focus. We have a couple of projects looking at both yield and quality in tandem, but there's not an example to which I can specifically point you at this time. I would say it's an area for active expansion that we are working on.
Yeah, and that's very much a focus on the animal side, that we have to look at multiple traits, because there's not a single trait that is overwhelming in terms of importance. I think we're going to have to leave it here. We're out of time. So I want to thank Christine for her presentation and participating, and all of the people attending and being involved in the polls and the discussion. We can continue the discussion on the Slack channel that's in the chat. Also, please fill out the survey. If you have ideas for future field days or want to share your work, please let us know. So our next field day would be the third Wednesday of May at the same time as normal. And we do have still some seed grants open, or call for seed grants is open, specifically the rolling and the community event grants and working group grants. They will close in April by the end of, well, applications close by the end of April next year, or until all funds are committed. So first come, first serve, assuming it's a proposal that is worth funding. So with that, thank you all, and have a good rest of the day.